I think we'll get started in a moment. Well, I have the pleasure to introduce Abigail Schindler, who we call Abby. And I've known her since she was a graduate student in Charlie Chafkin's lab, and she ended up spending about 6 months. Well, I got to know Abby over a period of time when she came and did some collaborative work with my lab. That was probably the best paper I'll ever have. And so I really appreciate that. And what's unique about Abby is her diverse interests in mechanism of basic neuroscience, fundamental changes in brain structure and function, how that changes the ability to make good decisions and to make wise behaviors, and also how that's impacted by clinically important problems like traumatic brain injury and PTSD. And so she really is a translational neuroscientist, and we need more people like her in our fields. We're lucky that she's staying here in Seattle. And she's progressed from a graduate student with Charlie in the pharmacology department to a postdoc with Jeremy Clark in the psychiatry department, where she worked up at the 15th floor and 16th floor at Harvard, at the university, on alcohol and juvenile development and decision-making and dopamine release. And now she is transitioning over to the VA as an acting assistant professor. She's going to be a great mentor, a great colleague now rather than a trainee. And soon a great, somebody who will train the next generation, which hopefully I will be able to watch and remember who she is later on, and my own dementia won't have proceeded too far. I think while she won't be talking about today too, Abby has a lot of diverse interests. She's also very interested in policy, science policy, and just has a very active and lively way of looking at problems. And I want you to join me in welcoming her to give grand rounds today.
Hello, everyone. Can you all hear me if I talk about this level? Perfect. Thank you, John, for that wonderful introduction. I'm really excited to be here and share some of the work that we've been doing over the past couple years. So as John said, I transitioned full-time to the VA about a year ago, so in mid-May of last year. But this collaboration really started in the end of 2014. So I'm hoping to kind of give you a background and then some of the newer data that we've been producing, and then kind of where I'm thinking of going in the future. Okay, and so first I want to start with talking a little bit about this multidisciplinary how we're approaching translational research in our group at the VA. And so you can see, let's see, so I am funded right now primarily through a career development award through the VA, and my mentors are listed here, with David Cook being my primary mentor, and then Elaine Peskin, and now Paul Phillips has taken over for Jeremy Clark in the role of mentor on the grant. And then here I have the help of Aaron Cooper, who's a wonderful lab technician, and then also some really talented undergraduates who have been doing some very tedious video analyses, where some data I'll show you later today, but they've been a huge help. And then a whole host of collaborators from the basic science and the clinical science side. And one of the really, I think, exciting things about the VA, or our group at the VA, is we're really able to do translational research, where I think this figure does a good job, where we're going from human patient populations to our animal models, back and forth. We're talking to clinicians, they're talking to us. And so we're really able to learn from each other, and what I think is in a way that has the potential to produce some really interesting results and therapeutics. Okay, so my talk, as John said, is going to focus on blast, traumatic brain injury, and PTSD, which it has I don't even know now, emerging issue. I think it's an established issue now. You can see that there's about over 2.5 million military personnel who have experienced this blast exposure. I'll get a little bit more into the detail of what the blast exposure is in a second. And this means that there is about 18% of combat deployed veterans return with a mild TBI. So not only are there a large number of people being exposed to blasts, there's also, of those, a large number is in this mild form. And then there's also this big comorbidity between mTBI and PTSD. which is something I'm really interested in, as I'll get into. A lot of the outcomes or symptoms overlap between the two. And so I've become really interested in trying to figure out what is from the actual physical injury of the traumatic brain injury, what is from more of a psychological component of the stressful experience, and what is kind of a synergy between the two. With the idea that if we can figure out some of these things, we may be able to devise therapeutics that target specific parts of the sequelae that results from mTBI/PTSD. And due to these facts, these events have become known as invisible rooms of the Iraq and Afghanistan war. And I think so on the right here, this is from a really, really great write-up, a National Geographic write-up. And these are masks that military personnel created once they've returned and are now in the VA system or seeking treatment. And I thought this one, looking at TBI and PTSD, just really spoke to the nature of these things that are happening and what the veterans are experiencing when they're returning to civilian life. And I do want to just say, I know on the original slide, I had a picture of a football player also. And so I'm going to be focusing on blast traumatic brain injury, but keep in mind that some of the pathology, some of the behavioral outcomes we see are very similar to what we're now seeing in some of the professional athletes. So I think this research has larger implications than only the VA or only veterans that have had this blast exposure. Okay, so why are these blast TBIs so prevalent in this population? I think this example here on the bottom, this is from just 1 year of a Stryker brigade in Iraq. And you can see that they had, 3,000 enemy attacks, 1,300 IEDs. That's just a year. A lot of military personnel are going over multiple mandatory deployments, and because of improvements in body armor, when they're now getting a blast exposure, they're able to basically get back up and keep going in whatever they were doing. And so they're also getting repetitive traumatic brain injuries or repetitive blast exposures. And so while, again, these are mild in nature, this repetitive nature ends up building up to have some pretty nasty consequences. Oh, and I was going to say, please interrupt me if you have any questions. I'd love for this to be a conversation, and then we can also have questions at the end. Okay, so for the blast injury producing forces, what we're going to focus on is this primary blast overpressure. And so this is the pressure wave that's created from this blast explosion. The secondary and tertiary, we're not going to focus on. That's debris and then the body being thrown against a wall, because a lot of times in these mild exposures and because of the body armor, those aren't as big of an issue in the mild blast exposure realm. And so this is a video of, you can see in Iraq from 2007. And what we're going to see is a car go around this corner, and then there's going to be an explosion. And I want you to pay attention to this area right here. We're actually going to see the ground change color, and that's actually what you're seeing is the pressure wave moving forward towards where the camera is. And know that there is someone standing back here by the camera that was able to get up and continue with their day. So there's the explosion. And then now you see, if you look right here, this is the pressure, the front of the pressure wave you see coming in. And again, there was someone that was able to, standing by the camera, get back up and continue. So now imagine that you had a bunch of these over the course of your military career. What kind of symptoms and outcomes are resulting? So the first are these somatic symptoms. And so I've broken these into somatic and then psychological or cognitive, but that there is a fair amount of overlap between these two. But common somatic symptoms are things like headache or migraine, light sensitivity, sensory motor integration disturbances, and then a big thing that I have heard the clinicians talk about, where they say the veterans are saying that the sleep disturbances is really decreasing their quality of life. Then kind of on the other side, these cognitive or psychological, and this is really how I got interested in this project. When I was doing my postdoc, there was a couple news articles about football and then about blast TBI, and we're looking at the symptoms, and I'm like, okay, aggression, impulsivity, substance use, these all seem like things that I'm already studying in a very different model, and things that are potentially partly mediated by the dopamine system. And that was the neurotransmitter, neuromodulator system that I was studying for my postdoc. And so that's when I approached David Cook and said, hey, I think we could potentially do some stuff together. And that's kind of how this collaboration started. So first, I'm going to take you through some work that has already been published last year. And so this was the initial research that I did with Dr. Cook and Dr. Peskin in the collaboration. And so this is looking at a clinical cohort of veterans. And so we have deployed controls, and then veterans that have gone through a blast-related mTBI. And you can see there's no difference in age or education or demographics. Here we're looking at the blast number. And so first off, you can see that the median blast number is 9. Some people were reporting greater than 100. So there's, this is, when I say repetitive, they mean repetitive. There's also at least a median of 2 with a loss of consciousness. I will say to be in a mild TBI, they have to have a loss of consciousness of less than 30 minutes.
Just to clarify, they don't have to have loss of consciousness. It's loss or alteration of consciousness. But if they have loss of consciousness, they have to be less than 30.
Yes, thank you.
And mostly it's less than that.
Okay, and that was Dr. Peskin saying that they don't have to have a loss of consciousness, but if they do, it has to be less than 30 minutes. So, okay, and then now looking at some of the behavioral and neurological measures. The first is looking at this NSI, so this is looking at some of those more somatic type of symptoms that I was talking about, and so you can see this is extremely significantly increased in our blast-exposed veterans. And I think that it's nice to have these deployed controls, because that's a control for it's not that it's something about going over and being involved in a active military. This is something about the blast exposure itself. We have a large increase in their PTSD score, and then also in the depression score. So when I first started, I talked to Dr. Peskin. I remember one of our first conversations, I said, okay, what are some of the top things that military personnel are talking about that's decreasing their quality of life? And some of the biggest things that she said was impulsivity and aggression. And so impulsivity was something that I was studying in my postdoc. So that was where I thought would be a good place to start. I already knew how to do a lot of these behaviors and was well-versed in the literature. And so this is looking at a behavioral disinhibition score that is a self-report that the veterans filled out once they're coming back and now at the VA. And so on the y-axis, we have the mean disinhibition, and you can see that we have a significantly increased disinhibition, self-reported disinhibition by the blast-exposed veterans. This is now breaking this disinhibition score into an individual question. And so this question is engages in risky behavior, and then it's a Likert scale. And you can see that the blast-exposed veterans have a significant shift over to the right, where you have almost 10% reporting that they almost always engage in risky behaviors, which is, if you think about it, risky behaviors can be a variety of things. A lot of them map onto some of those psychological things like substance abuse, risk-taking, and even aggression you could potentially look at. And so I think this is a really big shift. Finally, this is looking at their disinhibition score and the time elapsed since the last blast until the evaluation. And so we see also that up here, these clinically significant disinhibition scores are even out to 10 years following the last blast till evaluation. They're still having this significant increase in disinhibition. And so what this is suggesting is that this problem happens, or this type of behavior is produced, and then it doesn't go away, at least looking out to this 10 year time point.
With your deployed controls, is there a possibility that there are folks that are more risk averse that figured out ways to get jobs and things that they just weren't going to be in that situation, and the other folks that got into more blasts were more want to get into the action?
Yeah, that's a great question. And
I can answer that.
okay.
So deployed controls, about half of them are combat military specialties who got lucky and didn't have a blast exposure severe enough to cause symptoms consistent with mTBI, and the other half are administrative. And I think it's more of what you get assigned to rather than what you try to avoid. I think the Army doesn't really, the armed services don't really allow you to be quite that selective about what your job is.
And just for the people in other locations, so the question was about whether there's some kind of self-selection between the people that would go on to experience a blast. And Dr. Peskin explained that for the most part, no, it's not a self-selection. And in the deployed controls, there's quite a few that have seen military exposure, but just have not had a blast exposure. Yes.
Just a small follow-up to that question. How were they recruited to the controls?
This was.
Okay. So, well, this is my VA merit review grant. So, they're recruited identically. They're recruited via the VA clinics, via flyers at the VA. They're all from the VA. So they're all veterans receiving care at the VA. Would have seen the flyer, or been referred by a clinician. The two groups are very similarly recruited.
Okay, and I will get into this more later when I'm talking about the animal research that I'm doing. One of the things that I've started doing and will continue, because I'm really interested if there's any kind of baseline individual differences that then end up forming different trajectories following a blast exposure. And so I do baseline testing on all of my animals, and then I make sure that there's equal numbers. Say if some animals have a slight increase in impulsivity, I'm going to normalize across the groups for that. And then in that way, they start equal, but then we can also map on once I get a large enough data set to see if there are, say this group started more impulsive, maybe they go on to be even more impulsive or not. So give me, a couple years. Okay, so that kind of wraps up what was in that paper for the clinical set. So now I'm going to dive pretty much only into the rodent research. And so again, please feel free to ask questions about any of this. And so the first is, can we model what's happening in these wards? Can we model a blast mTBI? And I think the way that, a lot of the blast modeling, the construction of the blast tube happened before I became involved in this project. And I think how they went about this, how Dave and Elaine and James and their collaborators went about this, I think is just really amazing and genius. And I can't wait for this paper to come out. But essentially what happened is they interviewed blast exposed veterans, and Elaine, feel free to jump in at any point, but did very detailed interviews about their blast exposures, but then for their top 5 worst blast exposures, very, very detailed, to the point of, how far away were you? Were there tamping forces? So was this under a bridge? Was it in a backpack? That kind of thing. And then they worked with blast physicists to pull out what they thought the PSI and the impulse, which is PSI times second or milliseconds, what those parameters were. And then kind of came up with and so you can see each of these little triangles is one blast event. And came up with kind of this sweet spot of what the blast parameters are for a mild traumatic brain injury that people are experiencing in Iraq and Afghanistan. Do you want to add anything, Elaine?
Well, I think that's pretty, as you explained it. But what's really great is now we have over 400 of these individually evaluated blast exposures. And we're working with Ray Bennett, just retired from Baker Risk. He's a blast engineer who was on the team that designed our blast tube, and we're literally going to create a Bowen curve, like that curve that comes from the Israeli bus bombing literature, and you can see the lung fatality
Okay, and so as Lay mentioned, this is Baker Risk is the company that created our blast tube. This is a picture of the blast tube, and then here it is in the lab. This gentleman standing here is quite tall, so if that gives you an indication of how large this tube is. And so essentially how it works is we have an animal that's anesthetized in this exposure area, and they're put in a way so that the blast wave travels down and contacts their body in a very controlled, same way every single time. And over here in the driver and spool, we load helium to a high pressure, and then there's a mylar membrane that gets ruptured and causes that pressure to travel down the tube, the pressure wave, and then hit the animal or come in contact with the anesthetized animal. This is looking at an example in black of our pressure trace from our blast tube and then a TNT standard. And you can see we have very good correspondence between our tube and a standard. And you can see here we're getting at about an average of 19 to 21 PSI. That's where we're aiming for, and that's kind of just the high end of a mild TBI. Okay, and then just quickly, the methods that I'm gonna use, these are all male C57 Black 6. I'm really excited to start doing some females, but right now we have only males to talk about. These are all 10 to 12 weeks of age, which is, that's adulthood for a mouse. So I'm also interested in the future to look at if there's differences if I expose more somewhere in the late stages of adolescence, early adulthood, which is more closer, I think, to the age of the military personnel. The sham animals get the exact same anesthesia exposure, they just don't get the blast. So that's our control for anesthesia. And then I'll show you data either from a 1x blast exposure, so that's 1 blast on 1 day, or a 3x blast exposure, so that's 3 blasts, 1 per day over 3 days. And then finally, I'm doing a whole host of behavioral testing at baseline and then at specific time points following their blast or sham exposure out to 4 months post-blast as the farthest I've gone so far. But I think that's far enough to be in the chronic state. I'm not going to go into it any more than these next 2 slides, but I wanted to mention that in addition to all of this behavioral data that we have, the group at the VA has a lot of data that's also looking at neuroinflammation, neuropathology. For example, here is an image, and you can see these are 2 papers that have that are published so far with some of this data. And so this is showing a large increase in both phosphorylated tau and astrocytes GFAP staining following blast exposure. We also see blood-brain barrier disruption following blast exposure. This is something, both blood-brain barrier disruption and the neuroimmunology, something I'm really interested in exploring further, how that relates to some of these psychological behavioral outcomes that I'm seeing. So stay tuned for that also. But essentially what this is showing, we load in a fluorescent dye into the tail vein of the animal, and then this travels throughout the circulatory system. The dye is too small to escape an intact blood vessel, which is what you see here in the shams. But in the blast animals, because we have this blood-brain barrier disruption, you see that this dye is able to now get out of the vessel into the parenchyma. Okay, so that is the mTBI part. Now let's switch and say, can we model the traumatic stress part in this rodent model? So one of the things that a simple answer would be no, because animals are anesthetized when they're in the blast chamber. And I said, okay, that makes sense, until I started doing the blast exposures myself. And what I started to notice is on the 2nd and 3rd days of the repeated blast exposures, the blast animals, when they were put back in the isoflurane induction chamber, the anesthesia induction chamber, they were showing signs of stress, or what I thought looked like stress. And so from that, I thought, hey, it seems like there is some kind of, they're learning something, or they know that something bad is going to happen after they've been in this induction chamber for the 2nd time. So I wanted to explore that a little bit further. And so how I've gone about doing this is I've set up a paradigm that I'm calling blast-induced condition place aversion. So condition place aversion, condition place preference are methods that have been used for a long time. And it's essentially the idea that if you pair a specific environmental context with something appetitive, say cocaine, then if you give the animal a choice later of going into that context or another one, they're going to go spend more time in that one that was paired with cocaine. On the flip side, if you pair that with, say, something they don't like a drug like lithium chloride, for example, they'll spend less time in that environmental context paired with that aversive stimulus. So I'm using that paradigm with this blast exposure. So how this works is first the animals get a pretest. So this is a very simplified version of the CPA chamber. And so essentially it's 2 chambers. One side has vertical black and white stripes. One side has solid gray wall. So very distinct environmental contexts. For their pretest, they can go in both sides. And then there's also this middle chamber that's kind of just neutral chamber. Pretest, they go in both sides. We measure how much time. We make sure they don't have a bias for gray for some reason. Then they go through this sham and blast pairing. So in the morning, animals are confined to their sham morning box. And so say the vertical stripes. Then in the afternoon, the animals that are in the sham group will be confined to the other box, get a sham again. The blast animals will be confined to that other box, but then get a blast exposure immediately after. So they sit for 10 minutes in this environmental context, and then they go immediately into either sham or blast. Or really, it's immediately into the induction chamber, and then the blast animals will get a blast afterwards. And then finally, they get a post-test where they get to choose which area where they want to spend time in. And here, this is looking at the sham animals. So now I'm expressing this as a condition place aversion score. This is the amount of time they spent in the morning chamber subtracted from the amount of time they spent in the afternoon chamber. And so a negative number means they spend less time in the afternoon chamber, which is the blast chamber for the blast animals. And so you can see for the sham animals, they don't have any significant difference in the amount of time they spend within either chamber, which you would expect because they're getting sham exposure in both chambers. This is what the blast animals look like. So this -1 is their pretest. So you see that they start out with no significant preference for one box or the other. But over time, the earliest I've looked is 28 days post-sham or blast exposure. And so here at 28 days and out to 84 days, they're showing a significant condition place aversion to that environment that was paired with the blast exposure. And this is actually, these are repeated tests. So it's the same animal getting tested every time. And I think this is really striking that they're still showing such an aversion at day 84, because each of those post-tests can be thought of as an extinction session because they're not getting any blast or sham exposure. And so the fact that they're still showing a really big aversion, even after they've had extinction sessions, I think is also speaks to the power of this connection that they're forming. So here are just some other parameters I've collected looking at this. So this is looking at their feces output or their urine output when they're in those environmental contexts. And you can see the shams don't really have a difference between the morning and afternoon, whereas the blast animals in the afternoon, they have an significantly increased fecal and urine output, again suggesting that there's something different about this afternoon blast or this afternoon chamber than the morning sham chamber. And then finally, this is looking at their weight change correlated to their aversion score. And so over the 3 days of the repeated blast exposure, the animals lose weight. They're not feeling well. They're not eating as much. And what I found is that this is actually, the more aversion they show, the more weight they lose, or vice versa, or they're correlated, whereas the controls aren't correlated at all. So again, just adding more evidence that there is some sort of psychological stress component to this blast exposure. Finally, I wanted to just touch on some work we're doing with autonomic function. So this autonomic function is now, or has always been implicated in things like TBI and PTSD. And how we're looking at it is looking at their heart rate and heart rate variability. And so we're using a non-invasive approach to measure heart rate variability in these animals. They basically just stand on a platform and it gets their ECG in from their paws. And so what we see with this is that we see a significant decrease in heart rate variability following this blast exposure, which is very similar type of effects that are seen clinically in PTSD and or TBI subjects. So this is another avenue that I think is exciting and can be potentially a very simple therapeutic readout. So if we're able to change their heart rate variability with a therapeutic, what other kinds of things might we change also? Okay, so interim conclusions. The blast TBI and PTSD are large issues for the Iraq and Afghanistan wars that come with a host of both pathological and psychological outcomes. We can accurately mimic a lot of these parameters in our animal model. So now, my question is, can we establish a behavioral battery, behavioral and physiological battery that we can use? Then A, are we getting similar outcomes as what we're seeing clinically? And then if we do, then my plan is to use this behavioral battery as a way to test different therapeutics. Yes.
I was reading an article about personnel in artillery units and firing like rocket-propelled weapons being subject to repeated blast exposure, and I was just wondering, are we considering them as part of the TBI group?
No. So to be in the TBI group, you have to have had at least one blast exposure that was severe enough to cause acute symptoms consistent with mild TBI. So either have to lose consciousness or have alteration of consciousness in the realm of, dazed, confused, bell rung. And you have to have one to get to be eligible, but the average was in the mid-20s. This whole issue of military occupational exposure to blast, subconcussive blast, is a big issue now. I just went to a meeting that was all about military occupational exposure. We don't know what the effects are. There have been several papers written about effects in breachers who do, repeated blasts for, blowing open doors and such. And there have been definite symptoms that have, and really very objective stuff that have been found in breacher instructors, but not the breacher, the armed services personnel being trained.
Yeah.
So it remains a huge unknown. And there are also some other huge issues we haven't even touched on, and that is potential toxic exposures in the war zone from burn pits, insecticides, balmy 122-degree Baghdad days, high altitude in Afghanistan, Kabul is 6,600 feet.
Yeah.
So there's just a whole slew of issues.
Yeah, I was thinking of it in context of like the football players that, high school football players and college football players repeated, no major concussion, but repeated injuries.
Yeah, it's and like Elaine said, I think it's going to be increasingly active area of research.
There's been a postmortem diagnosis case of chronic traumatic encephalopathy in a 17-year-old high school football player.
Wow.
So these are giant issues in our society.
Yeah. Okay, so now we're going to switch over to looking at some of these behavioral outcomes that I've talked about. I'm going to talk about sensory motor integration and then really switch over to these more psychological outcomes. So focusing on impulsivity, anxiety, depression, aggression, and substance use. And then keep in mind that I don't have an arrow on PTSD, but a lot of these are part of a PTSD type of sequelae. So first, looking at sensory motor gating, this is something already published by Dr. Mebon. And so this is looking at the rotor rod. And so essentially they're looking at, this is a rod that starts to spin faster and faster and faster. And we look at how much time the animal stays on the rod, how well they can perform the task. And so with one blast exposure, we don't see any significant difference. But here, starting with 3 blast exposures, we see this significant deficit following blast exposure, 3 blast exposures. Another way to look at this is looking at the acoustic startle response. And so this is, the animal is enclosed in a kind of chamber, and then there's a pressure sensor underneath. And so when they do a startle, that pressure sensor is able to record that. And so we play a loud noise, an acoustic, and then they startle. And so first, this is looking at, so we have startle on the y-axis, and then increasing decibel levels of the noise that we're playing to create the startle. And this is a significant interaction. So now we're almost only going to show you 3x data from here on out, and mostly look at the 56 and 84 day time points. If anything, what I seem to be seeing is things are getting worse over time. I don't necessarily think it's because they're not there initially. I think it's because there's so much of the physiological part that doing some of these behaviors it just gets really messy. And so I think really for behaviorally looking starting 2 months is kind of the sweet spot.
And sort of as a side question, is there anything known either in humans or in this model about the actual sensory processing of sound that happens at that part of the spectrum?
As far as like hearing loss? Yeah. So that's right here. What this is suggesting is that we're seeing significant hearing loss. But it may be different at different decibel levels.
And in terms of what our veterans report and complain about is they have both.
Oh, okay.
Hearing loss and sensitivity to loud noise.
Interesting. Yeah.
And just sensitivity to noise
In general. Yeah. Yeah. The tinnitus, yeah. So because of this is because there's differences in hearing both at low and high levels. This isn't the best way to look at acoustic startle. So what we have now looked at is something called the Pre Pulse Inhibition. And so this is, you play a softer pre-pulse and then a larger pulse, and that pre-pulse should inhibit your startle to that larger pulse. And you can see in the control animals, they do. This is the amount that the pre-pulse inhibits the pulse. And so, and then this is looking at the PP pre-pulse delay, is how much time in between the pre-pulse and the pulse. And so basically you can see that these blast exposed animals have a deficit in pre-pulse inhibition. And that it especially is seen at these shorter delays between the pre-pulse and the pulse. And because this was normalized to the pulse, this at least takes out some of the issues with the hearing loss. We've also looking at startle habituation. So this is playing the same decibel level 50 times in a row. And so normal animals, control animals, you can see here that over those 50 trials, they habituate to that pulse or that noise. Our blast exposed animals do not habituate. If anything, at this 56-day time point, this is a positive slope, not significant yet, but if anything, it's looking at potentially they're sensitizing to this repeated decibel level. Okay, so now looking at novelty seeking. So this is novelty seeking is related to things like impulsivity, disinhibition, and risk taking. And so this was a initial kind of simple behavior to do to go coincide with the self-report disinhibition scales that we were looking at in the veterans. And so essentially what this is the animals are in an open cage, and then they get a novel object placed in the center of the cage. And so I'll show you a picture of them now. They're things like these that I get from Archie McPhee's. That's my favorite store for science experiments, actually. And so, for example, they'll have a rooster put in the middle of their cage. And then all we're doing is measuring the amount of time that they spend interacting with this object. And increased time interacting with the object is a increased novelty seeking. And so you can see this is at 7 days post-sham or blast, and then here we have 30 days post-sham or blast. And we see, and then this is the time they spend and then the entries into the novel object area. And so on both of these parameters at both time points, for both 1 and 3x blast, we see this significant increase in this novelty-seeking type of behavior. I'm almost set up to do operant behaviors now. Next month, I think we'll start some. And so we can do some more sophisticated type of behavioral assays to get more at impulsivity, impulsive action, impulsive choice, risk-taking. And so again, in the future, I'll have a larger, more full data set to talk about. Okay, and then so this is looking at over time now. So I showed you that was a 7 and a 28 day. Now we're going out to 84 days, and we're still seeing this increase, a significant increase across groups in the between sham and blast exposed animals. And I use just as an aside, I use a different novel object each time because they have, if they've seen it once, it's not novel anymore. So the guy really was like, why are you buying 5 of each of these barn animals, or 6, because we have 6 chambers, and he kind of got a kick out of what I was doing. Okay, so that's novelty seeking. Next is something called marble burying. So this is a test of anxiety and potentially OCD type of behaviors. And so essentially you get this cage with about 5 centimeters of bedding, so a large amount of bedding. You place the marbles on top, and then the animals just naturally want to bury the marbles. Animals that are in a heightened anxiety-like state or have a genetic predisposition for OCD type of behaviors will bury more marbles. So this is what this looks like with our blast or sham animals across time. You can see that the blast animals across all 3 of these time points have an increase in marble bearing. So this kind of anxiety-like behavior. And here you can see, again, this is their pre-test. They don't start with any different baseline differences. We've also looked at the elevated plus maze. So this is another test of anxiety / risk-taking. So essentially you let the animals travel throughout this maze, and these open arms are much more anxiety producing, or animals that are in a anxious or dysphoric type of state will spend most of their time in the closed arms. Animals that are risk-taking are more likely to spend time in the open arms. We were traditionally thinking of this as a test for anxiety, but if anything, it's showing us that the animals have an increase in risk-taking because they're spending almost double. It's hard to tell because of the closed arms, but they're spending almost double the amount of time in the open arms as the control animals. So this is, again, going in line with more of this novelty-seeking disinhibition type of behavior. So I think it's really interesting. As you can already tell, we're having kind of both sides of this behavioral outcomes. We're having anxiolytic type of things, but then also this risk-taking, novelty-seeking type of behavior. Okay, this is looking at a depression-like behavior. So this is a sucrose splash test. And so what this is a 10% sucrose solution that you spray on their dorsal fur, and it's sticky. They want to groom it off. Animals that are in a stress, like a chronic stress state or in a depressive-like state, will spend less time grooming. And that is what we see in our blast animals. We find a significant decrease in the time that they spend grooming following this administration of the sucrose solution. They also have a decrease in the grooming duration and in the number of grooming bouts that they see or that they exhibit. This is the data that my lovely undergraduates did by hand on the video. It's a lot of video analysis to do, so thank you undergraduates. Okay, final behavior is something called a tube dominance test. So this is getting at aggressive type of behavior or dominance type of behavior. And so what we have is a clear tube that is only big enough to allow one animal to go through. And you put an animal on, can't show you with my fingers, on each side. And then there's walls that you then will pull out, and then they meet, and one is gonna force the other one out of the tube. Normally the dominant or aggressive one will force the other one out of the tube. This is now expressed as wins per bout. And so what I do with this is I have a sham cage go against a blast cage in a round robin. And so if an animal wins every one, then they would have a score of 1. If they lose every bout, they'd have a score of 0. You can see at pre-test, we're at about a 0.5, meaning that they're equally balanced. Now, what I think is really, really fascinating is that initially at this 28-day time point, the sham animals are winning most of the time. Over time, now out to this 112-day time point, the blast animals are winning most of the time. And so there's some sort of transition from, I think this is partly the 28-day time point is partly because of they're just feeling like crap. They don't want to fight. They're just like, leave me alone. I'm getting out of this tube. Whereas by the end of the 112 days, they're not feeling as much of the physical part. And now they have this aggression psychological part potentially coming into play.
And this is within subject.
Yep. Yeah, and it's the same cage match every single time over time. I didn't believe this at first because I'm like, no way. And I did it with 2 more groups now. And so this has been replicated. So I'm really excited. We have a new aggression researcher joining the faculty at the University of Washington. And we've already been talking about collaborating with some stuff. He has some really cool models. So I'm excited to move farther with this. Okay, and then finally, for the last behavior, and I'm gonna go kind of fast, is now looking at ethanol intake. So substance use/misuse is a potential outcome of TBI and or PTSD and self-medication. And so this is looking at an intermittent 2-bottle choice paradigm. So this is the animals are getting to choose whether they want to drink alcohol or not. So there's 2 bottles in their home cage. They get it 24 hours a day. One has alcohol in it and one has water. So they get to choose. And then we're looking at how much ethanol that they're drinking per body weight per 24 hours. And we do this Monday, Wednesday, and Friday. It's an intermittent schedule, and we do it for 3 weeks. And so what you can see is that the blast animals they're drinking significantly more. This is their 24-hour data. If we look at just 2 hours into their dark cycle. So I put the alcohol on at about 5:30. Their lights go out at 6, which is their kind of daytime. And then I measure it again 2 hours in, so at 8 p.m. And so here you can see that there's a large increase in the amount the animals are drinking. 8 grams per kilogram in 2 hours is a pretty large amount of alcohol. I'm setting up video ability so we can actually look at what they're doing when they're drinking, which I think will be really interesting too. And then this is looking at their ethanol preference. And so, yeah, they may be drinking more alcohol, but maybe they're drinking more water also. And that doesn't seem to be the case. The blast exposed animals are preferring to drink the alcohol, whereas the sham animals are maybe a little bit over 0.5, but not by much.
Have you tried the MSNBC versus Fox?
Next week. Okay, and then I want to quickly go through the molecular mechanisms because I want a little bit of time for questions. But as I said in the start, and Dr. Neumeier talked about, my background is in the mesolimbic dopamine system. So this is what I studied during my postdoc. And I thought that this potentially plays a role in a lot of these behavioral outcomes we're looking at for a variety of reasons. One is that this system is linked to things like motivation, decision-making, risk-taking. There's an increase in dopamine in the nucleus, in phasic dopamine in the nucleus accumbens found in a lot of these behaviors that we're looking at. And then also dopamine is very intertwined with drug addiction or substance use and abuse. The way that we look at this in the lab is through a technique called electrochemical detection of fast-scan cyclic voltammetry, or FSCV. So essentially, this is a way to get sub-second resolution of phasic dopamine release in discrete brain regions while the animal is awake and performing a task. So this is a human hair right here, and this is the carbon fiber microelectrode that we implant into the brain. This is very small, obviously, so it's not doing damage to the brain tissue. We're able to, with stereotaxic coordinates, be very precise about what brain region we're targeting. So I target the nucleus accumbens core. And then essentially what FSCV does is take use of the fact that dopamine is electroactive, meaning it can be oxidized or reduced. And so we apply this triangle waveform potential to the carbon fiber, where when the potential is increasing, dopamine is oxidized, which means it's freeing up electrons. Those electrons are read by the carbon fiber, and it's directly proportional to the amount of dopamine. So the amount of current read by the fiber is directly proportional to the amount of dopamine that was oxidized. And we run this at 10 hertz, and so that's how we're getting sub-second resolution for the amount of dopamine. So right now, I'm only going to show you data from anesthetized animals. And so this is where I am implanting a stimulating electrode and then measuring dopamine release when the animal is anesthetized. Once these operant boxes are up and running, we will be able to do this in awake behaving animals. And so we'll get dopamine released to something like them pressing a lever or them seeing an alcohol cue, or a fear cue that was associated with the blast exposure, those kind of things. And the reason why we're targeting the PPT to start with is this sends cholinergic and glutamatergic excitatory afferents to the dopamine neurons in the BTA. And that this brain region is also really important for a lot of these behaviors that I've been talking about. So this is shown just some example color plots and traces. And so the blue is a control animal, and then the red is a blast exposed animal. And you can see that in this anesthetized stimulation prep, there is a significant increase in phasic dopamine release following blast exposure. This is, now you can see across stimulation currents and frequencies. This is looking at 30 days following blast exposure. And then here, I actually took it out to 120 days post-blast exposure, and we still see this significant increase in stimulated phasic dopamine release. So I'm really excited to start doing some of the awake behaving experiments, because right now, all we can say is when we stimulate this brain region, this is what we see. We don't know what's gonna happen when they actually start doing tasks. Okay, and then just to sum up everything, as I said, I think we're creating a really nice model of both the blast traumatic brain injury and the psychological stress or the trauma that happens. And then now I think we're getting a really nice physiological and behavioral battery where we can start testing potential therapeutics. And then again, I think that this is, as we've been talking about, this is a growing health concern, not just for military personnel, but for athletes, for just civilians at various events around the world. And so with that, I will take any questions. Thank you. Yes.
I'm curious, I don't know, do the glutamate electrodes that are out there have time resolution, or they work in vivo so you can see if there's a decrease, if there's a loss of inhibition in the BTA, for example, from cortical impulse?
Yeah, so from what I understand, although I'm not super up to date on that literature, but what I understand is it doesn't have the same time resolution yet. But I know there's technological advances all the time. I would love to collaborate with an electrophysiologist to look at some of this, if there's differences in the dopamine neurons or the GABA neurons of the BTA. Because yeah, if we do see this increase in dopamine release, we don't know what the mechanism is, and it could be a variety of things. Thank you.